Hello and welcome to the present lecture on the topic of evolution of visceral arches. Visceral arches, also known as pharyngeal arches, are a series of five paired swellings. These swellings may be seen in the developing embryo around 30 days of development. These arches are numbered as 1, 2, 3, 4, and 6 in case of human beings. The violet colored series of these swellings have been shown in this slide as gill slits. These are actually the developing pharyngeal arches. The pharyngeal arches are unique to vertebrates, and these are these bilateral paired swellings that surround the foregut of the embryo. These are formed by the cells from ectoderm, endoderm, mesoderm, and neural crest. Ectoderm lines the external surface of these pharyngeal arches. And as you can see in the slide, the ectodermally lined depressions on outer side of these pharyngeal arches, these are called pharyngeal grooves or pharyngeal clefts. Whereas the endoderm lines the internal surface of these pharyngeal arches, and the depressions formed on the internal side, these are called pharyngeal pouches. The ectoderm in the slide has been shown as blue, whereas the endoderm has been shown by red color. The section of these pharyngeal arches show that the center has been filled with mesodermal mesenchyme. That is mesoderm, and the mesenchyme from the neural crest cells that lies at the periphery of this central mesodermal mesenchyme. The arches have been numbered as first, second, third, fourth, and sixth. The fifth one is missing in case of human beings. You can see in the slide here on the right side, the blue lined ectodermal clefts or grooves that have been numbered as first, second, third, and fourth. That is, the first lies between first and second gill arch, and the second groove or the second cleft lies between second and third. Similarly, you can see endodermal pouches that are being lined with a red color on the inner side. They are also numbered as first, second, third, and fourth, lying between the first, second, or the second, third these pharyngeal arches. The slide here shows the components are the structures in a pharyngeal arc. A typical pharyngeal arc, it contains an aortic arch, that is an artery that arises from the truncus arteriosus of the initial heart developed at that time. Second is a muscular component that you can see that differentiates into the muscles in the head and neck region. You can see a nerve that supplies the mucosa or the muscles derived from the arch. And you can also see a cartilaginous rod or the cartilage that forms the skeleton of the arch. This is the main thing that we will be focusing in case of these visceral arches, and that is these cartilaginous rods. These cartilaginous rods later on led to the development of the skeleton of the facial region, that is the upper jaw, that is the lower jaw, and the anterior neck region, as well as we can be seeing these things in the coming slides. These cartilaginous rods are the skeletal elements. These are the final structures to develop in each of the pharyngeal arches. Before we proceed to discuss the skeletal derivatives, that is, the skeleton or the bony structures found from these cartilaginous components of these visceral arches, we will be first taking the fate of these ectodermal clefts or grooves and these endodermal pouches. In simple terms, we can say that the structures that are developed from these ectodermal clefts or from these endodermal pouches. We will be first having a glimpse of these, and then we will proceed finally to the skeletal derivatives of these visceral arches. We will not cover the fate of these ectodermal clefts or endodermal pouches in detail. Like you can see in the slide here, the first cleft on the ectodermal side, on the outer side, it leads to the formation of these external acoustic meatus of the ear and external part of the pinna. Like the ventral part of it is obliterated. Uh, Similarly, you can see in the next slide here, these endodermal pouches is having different fates, like the first one forms tubotympanic recess, the second one allows the formation of this tonsil, the parathyroid, and the lateral thyroid, thymus, etc. All these are the different endodermal pouch derivatives that are later on developed from these pouches. Similarly, in this slide, you can see the nerves and muscles of these pharyngeal arches. Like the first arch is having being innervated by the mandibular nerve, and the number of muscles like the tensor tympani, like the tensor palatii, etc. These are formed from this first arch. And similarly, the second arch, for example, is innervated by the facial nerve, and there are the number of muscles related to the facial expressions. These are found from the mesodermal component of this second arch, and similarly the other arches as well. Now the skeletal derivatives in case of human beings. Here you can see the derivatives of first visceral arch have been shown in red, whereas the derivatives of second skeletal elements have been shown in green, and third yellow, and the fourth and sixth they have been shown in white. You can see in red derivatives, skeletal derivatives of the first visceral arch, like the incus and malleus bones of the internal ear, the ligament that is mandibular ligament, like the remnants of Meckel's cartilage within mandible, they all have been derived from this first visceral arch. Similarly, from second visceral arch, you can see here styloid process of the temporal bone, styloid ligament, the upper part of the body of this hyoid bone, lesser cornua that has been shown in green. They are derived as the skeletal elements of the second visceral arch. The third visceral arch, you can see the lower part of the body of this hyoid bone, the greater cornua of this hyoid bone, they have been derived from the third visceral arch. Similarly, fourth and sixth visceral arch led to the formation of this thyroid cartilage and the cercoid cartilage. So it shows how these different visceral arches led to the formation of the different skeletal elements of the facial and neck region. Hence, we see the first arch will develop into maxillary and mandibular swellings, and this will give rise to mid face and lower face, respectively. The first arch cartilage in the maxillary swelling will form the incus and the aliciphenoid. The principal cartilage of the first arch is a rod in the mandibular swelling, that is Meckel's cartilage. This forms the malleus and the two ligaments, that is the sphenomandibular and the anterior malleolar ligaments. The remainder of the Meckel's cartilage disappears. The second arch cartilage forms the stapes, the styloid process, the stylohyoid ligament, and the lesser horns of the hyoid bone. The third arch cartilage contributes to the hyoid body and the greater horns of the hyoid. The fourth and sixth arch cartilages contributed to the laryngeal cartilages. This was the case of humans, where only five visceral arches are found. Generally, arches number varies from 4 to 9 in different animal groups. Like in fishes, there are 7 visceral arches. The first visceral arch, as you can see in the figure here, it's called mandibular arch. Second one is called hyoid arch. And then from third to seventh visceral arches, they are called branchial arches. First and second visceral arches are related to the formation of these facial regions, that is jaws, and then the anterior neck regions, as well as the attachment of jaws with the uh, cranium. Whereas from third to seventh visceral arches, they are the branchial arches, they are linked with gills here. That is, they support gills, and the cases varies in different groups of fishes. We will come to it, but first let's see the first arch, that is mandibular arch. It is made up of mandible and palatal-quadrate components. Similarly, there is hyoid arch that is made up of hyomandibular, epihyal, that is epihyal, ceratohyal, and basiohyal, and the branchial arches. They are made up of four pieces of cartilage, namely pharyngobranchial, epibranchial, ceratobranchial, hypobranchial. We will be seeing at the end of this lecture a table in which it will be mentioned how different parts of these different arches led to the formation of different skeletal elements or different structures. These visceral arches show diversity or variation in terms of number, in terms of the function of different parts in different vertebrate groups. However, some groups may show similarity in some cases as well. Like we see in cyclostomes, that are jawless fishes, jawless pisces. They possess a splachnocranium in which usual cartilages are not identifiable. What is this splachnocranium? Actually, those visceral arches which contribute to the formation of the skull constitutes the splachnocranium. In cyclostomes, the whole pharyngeal skeleton, that is this visceral arches skeleton, this fuses to form branchial basket to support gills. As there are no jaws, all these arches or cartilages are involved in the formation of the branchial basket, and that is the support to gills. In elasmobranchs, that is the cartilaginous fishes, they contain full set of visceral arches and three unpaired branchial cartilages called basibranchials. They have five pairs of functional gills, and these gills are supported by these skeletal structures of visceral arches. In case of bony fishes, Meckel's cartilage forms articular and becomes part of the lower jaw. What is this Meckel's cartilage? Actually, the cartilage of the first visceral arch is given the name Meckel's cartilage, and the cartilage of the second visceral arch is given the name Rettach's cartilage. These are the only two cartilages which are being given names, and the cartilages of the rest of the branchial arches are not given any special names. So the Meckel's cartilage is the cartilage of the first visceral arch. Meckel's cartilage forms articular. What is this articular? Articular, actually, you will see in jaw suspension, a bone which helps in the articulation of the lower jaw. It becomes part of the lower jaw. And hyoid arc, that is the second arch, is modified for the movement of the operculum and functioning of the lower jaw. Similarly, the last branchial arch in case of these bony fishes, it shows sign of degeneration. So the number of gills is reduced to 4 from 5. Like in amphibians, larval frogs have 6 visceral arches and the last 3 bear gills only. Similarly, reptiles, birds, they also show variation in terms of the number as well as the structures derived from these branchial arches or visceral arches, or the support given to the different organs or different elements surrounding a pharyngeal region. Now the table in this slide, it shows the derivatives of different branchial arches in case of sharks, bony fishes, and the tetrapoda, that is amphibians, reptiles, birds, and mammals. Different colors have been shown to differentiate between the different derivatives from different parts of these visceral arches. Like the first visceral arch, Meckel's cartilage, it gives rise to articular in case of the teleosts, amphibians, reptiles, birds, but not in case of mammals. In case of mammals, this gives rise to malleus only, that is the bone of the internal ear. Like the palato-quadrate part, it forms quadrate epipterygoid in case of teleosts, amphibians, reptiles, and birds, but not in case of mammals. In case of mammals, it forms incus and aliciphenoid. To understand our To comprehend these different branchial arch derivatives, it is necessary for us to have the understanding of the skeleton of any of the vertebrate groups. For example, I have given the description link. Sorry, I have given the link of the rabbit skull endoskeleton in the description below. You can just see that, see the video to understand the different skeletal elements of the endoskeleton of this rabbit skull. From there, we can have an idea what is this quadrate, what is this abipterygoid, or what are these different bones that have been shown here. Also, the derivatives are the skeletal elements like the styloid process of the temporal bone, the body of the hyoid, or the second horn of the hyoid. I In the coming slides I will be showing some bones from where you can get an idea that what are the structural elements derived from these visceral arches. I haven't given the full description of this table. Rather, you can pause the video here and you can see the table for all the structural or the skeletal derivatives of these different visceral arches in case of fishes or in case of tetrapoda, that is amphibians, reptiles, birds, and mammals. To understand it fully, already told that there is a link in the description below. You can first visit that, and then it is possible for you to understand this table. And now this is the temporal bone of the humans. Here you can see styloid process. This styloid process has been derived from one of the visceral arches. Now this is the hyoid bone. You can see the different parts of this hyoid bone like body, the horns of hyoid bone. I hope the lecture might be of some help for you.
PHANKYOU. Thank you. Jazakallah.